Welcome back to my channel Knowledge Chronicle.
Imagine this. You're made up of trillions of cells, and inside every one of them is a tiny instruction manual that decides how you look, grow, and even how long you might live. But what exactly writes those instructions? Is it protein, DNA, or something else entirely? Today, we dive into one of the greatest scientific mysteries ever solved: the search for genetic material. Stay with me till the end, because what you'll learn today will forever change how you see life, inheritance, and evolution itself. A mystery begins. In the mid-1800s, two powerful scientific events were taking place. On one hand, Gregor Mendel was discovering the basic laws of inheritance using pea plants. On the other, Friedrich Miescher isolated a mysterious substance from the nucleus of white blood cells. He called it nuclein. But while Mendel laid the foundation for genetics, no one knew what this nuclein really was. Was it just another cellular chemical, or something much bigger? By 1926, scientists knew that the secret to inheritance was hidden in chromosomes inside the nucleus. But what part of the chromosome carried this blueprint? Proteins, or nucleic acids like DNA. Griffith's transforming principle. The first big clue came in 1928 when a British scientist named Frederick Griffith experimented with two types of Streptococcus pneumonia bacteria S strain smooth, with a polysaccharide coat that made it virulent deadly, R strain rough, without the coat non-virulent. Griffith injected these into mice. Mice with S strain died. Mice with R strain lived. Heat killed S strain, mice lived. But the twist: heat killed S strain plus live R strain equals sign mice died. And from the dead mice, Griffith recovered live S strain bacteria. What just happened? A transforming principle from the dead S strain had turned the harmless R strain deadly. But what was this mysterious substance? Avery, MacLeod, and McCarty. DNA takes the stage. In the 1940s, three scientists, Oswald Avery, Colin MacLeod, and Maclean McCarty, took Griffith's experiment a step further. They broke open heat-killed S cells and isolated different biomolecules, proteins, RNA, and DNA. Then, they tested each to see which one could transform R cells into S cells. Results? Destroying proteins, still transformed. Destroying RNA, still transformed. But destroying DNA, no transformation. That was big. They concluded that DNA must be the genetic material, but not everyone was convinced. Many still believed proteins were too complex not to be the carriers of inheritance. Hershey and Chase, the final blow to protein theory. Enter 1952. Scientists Alfred Hershey and Martha Chase devised a now legendary experiment using bacteriophages, viruses that infect bacteria. Here's what they did. They labeled DNA with radioactive phosphorus P32. They labeled proteins with radioactive sulfur S35. Then they let the phages infect E. coli bacteria. After infection, they shook off the viral coats and spun the samples in a centrifuge. The results? Radioactive DNA entered the bacteria. Radioactive protein did not. It's the DNA that carries genetic instructions, not protein. Case closed. DNA is the genetic material. Why not RNA or protein? So why DNA? Why not RNA or protein? Let's break it down. To be a genetic material, a molecule must 1. Replicate itself, must make accurate copies. 2. Be stable, can't break down easily. 3. Mutate slowly, to allow evolution. 4. Express traits, code for proteins like Mendel's characters. Proteins, too unstable and can't replicate. RNA can replicate and mutate, but it's too reactive and breaks down quickly. DNA, on the other hand, has a double helix structure. Base pairing rules ensure accurate copying. Lacks a reactive OH group unlike RNA, making it chemically stable. Has thymine instead of uracil, giving it added stability. Mutates slowly, allowing evolution without chaos. Even Griffith's experiment hinted at DNA stability. The heat-killed S bacteria still had functional genetic material. Interesting twist. In some viruses like tobacco mosaic virus and QB phage, RNA is the genetic material. These are exceptions, and they tend to evolve much faster. So both DNA and RNA can serve as genetic material, but DNA is the king, more stable, more precise, and perfect for long-term storage. Let's pause for a common question. What's the difference between DNA and DNase? DNA is the genetic material, a molecule made of nucleotides that carries instructions. DNase is an enzyme. It breaks down DNA. When Avery and his team added DNase to the transforming mixture, it destroyed DNA and transformation didn't occur. That proved DNA was essential. So let's wrap up the journey. Mendel gave us inheritance. Miescher found nuclein. Griffith discovered transformation. Avery and team pinpointed DNA as the transforming factor. Hershey and Chase delivered the final proof. And today, we know DNA is not just a molecule. It's the script of life, passed from one generation to the next, shaping everything we are. Next time you look in the mirror, remember, you are a walking, talking collection of instructions written in DNA. If you found this story as fascinating as we did, don't forget, your one like, share, and subscribe will help us create more valuable content. Until next time, stay curious with Knowledge Chronicle.